A very good evening to everyone. Once again, we welcome you all for Drug Discovery Hackathon 2020 training program, Vertical 3 session. In this session, we have eminent scientist Dr. B. Jayaram, Professor, Indian Institute of Technology, Delhi. Dr. Jayaram is the faculty of Department of Chemistry in IIT Delhi since 1990. He is a recipient of various awards, including CRSI Medal and IBM Faculty Award. He is the principal investigator and coordinator of the Supercomputing Facility for Bioinformatics and Computational Biology in IIT Delhi. And his group is mainly responsible for developing Chem Genome, Bhagirath, and Sanjeevani methods, and software for genome annotation, protein tertiary structure prediction, respectively. And his group also responsible for setting up a multi-teraflop supercomputing facility for bioinformatics and computational biology at IIT Delhi. And his group also mainly making the hardware and software facility freely available to the global user community. And Professor Jayaram is also a founder director for 2 startup companies formed by students and project personnel of SEH Bio, IIT Delhi. With this small introduction, I request Dr. B. Jayaram to deliver the lecture on integrating chemistry with biology and IT towards a disease-free planet. Sir, please, sir.
Oh, thank you very much. At the outset, I would like to thank Professor Shastri and his team for arranging this. So, good evening to all the participants. I cannot say I'm happy to see you all, but probably I do get to see you virtually, but someday we'll catch up in person. So today my show will be on integrating chemistry with biology and IT. As you know, these 3 are the emerging disciplines, especially for drug discovery. And our main goals is someday we'll have this planet free of all diseases. Well, if not in my generation, in your generation, definitely this seems feasible. So what is the motivation for this aspiration? Well, the motivation is very simple. We are living the world of big data, and this data is increasing tremendously. And when I say big data, one of them could be the genomic information of several thousands of organisms, and protein sequence informations in millions and millions of entries, and the protein structural information, structural data banks are swelling. Then small molecules, this seems like a little dated, but small molecules, again, 35 million purchasable compounds. And so when we're talking of data that combines genomes, proteomes, and small molecules, probably hundreds of petabytes is an underestimate. And when you add PPI networks, gene expression, maybe we're dealing with exabytes. What do we do with all this data? Well, the hope we can probably use some of this data and make some drug molecules and get rid of diseases from the planet. So that's the overall big dream. Now, another idea, motivation here is not only coming up with products which are useful to society directly, but it could also be used for generating new knowledge. So from big data, you go to information to knowledge. So this in itself is a laudable goal. Then, of course, utilizing this knowledge, we're going towards the products useful to society. So that's a big challenge today. Now, what it requires is hypothesis generation. How do you generate a hypothesis using the data? How do you validate it eventually to create new knowledge? So these are some interesting questions, especially in the context of genomic, proteomic information and the small molecule databases that we have. So move on. And my dream, of course, is that if you have a genome card, someday I'm sure all of us will carry an identity card, which probably has a genome coded in it, our own genome coded in it, right? So at 10 a.m. the disease is reported on a smartphone along with a genome card and other reports and symptoms. 5 p.m. the drug is delivered at a doorstep, whatever the disease. So that's the dream. Can we do it? Well, one simple pathway that I am envisaging here that I'm sharing with you is that you start with a genome, then move on to this protein space, and then create a 3D structure, then scan molecules against these binding pockets, and eventually come up with an inhibitor molecule. And then hopefully send the lead molecule to the pharmaceutical for synthesis, clinical trials, approvals, etc., recommendations, and then administer to the patient, and the patient is happy, running, smiling, jogging, etc. So is this conceivable? Probably yes, and maybe in 50 years from now, this will be a routine matter. So I don't think we should be worried too much about diseases 50 years from now, but today, of course, we are worried, and maybe we have to contribute towards this goal. And what will be the tools that we will have to use? Well, probably genomics, proteomics, metabolomics, information technology, and chemistry. If we can somehow automate all this, then it will be a wonderful scenario for the new world that we are yet to see. Now, what's the urgency for drug discovery apart from the current pandemic? Look at the discovery of these antibacterials. So what we are seeing is that up till mid-80s, there was a ferment activity, and several molecules have been discovered. And beyond that, and you start to see a discovery void. You don't find too many molecules. And on top of it, that means we are very complacent. We think we have conquered the bacteria, and bacteria are not a problem anymore. On the other hand, bacteria are becoming smarter in the sense that this antimicrobial resistance is on the increase. Now imagine a scenario where the AMR takes a peak. Then what would happen? And none of the antibiotics is working. Then it doesn't have to be a deadly disease or something very complicated. It would just fall from a bicycle. A bicycle can be a killer disease. Fall from a bicycle can be a killer disease. So that could be a scenario which we don't want, and we hope we will come up with some new molecules here. So that's one reason why we want to be continuing the drug discovery, especially with newer data and newer technologies and newer methodologies. Now, some milestones in drug discovery, as I put it together, I'm sure everyone has a different perspective here, right? And one of the Nobels was for this antibacterial molecule Prontosil. That was way back in 1939. Then 1945, you have the discovery of penicillin, a well celebrated the discovery. I'm sure all of you know the serendipitous discovery. Then move on to the structure of the penicillin, thanks to Professor Hodgkin, and for solving the crystal structure and telling us about this beta-lactam. Apart from this, she also did the vitamin B12, and simultaneously, we have a streptomycin, which again is a very powerful antibacterial compound, and this is again celebrated in 1952 with the Nobel. Then more recently, a few years ago, I'm sure all of you heard of this artemisinin for malaria, and ivermectin for the blindness and lymphatic filariasis, and this was again the Nobel in 2015. So then question then is, well, these are all nice, good stories, and society is thankful to all these inventors. How do you design a drug? Well, that's what is captured in this the Nobel citation here, that you focus on the biochemical pathway, take a rational approach, take a basic biochemical physiological process. And today, of course, we are inspired by the structure. So we say we go one step beyond the physiological process. We say, well, let's just get the structures as well, because structure dictates function. And then a malfunction is what is causing the disease. So can we intercept the issue at the structure level? So that's how you start creating a logic, a framework, a fabric for structure-based drug discovery, taking off from this 1988 Nobel. And now you would ask, so why is this ribosome interesting? Apart from the technological feat that it celebrates, so many atoms, hundreds of thousands of atoms captured through X-ray crystallography by these three famous scientists, it's also a good drug target for coming up with this new antibacterial, new generation of antibacterial. So that is one more motivation as to why one would like to have a structure of ribosome, and Venki Ramakrishnan, Stites, and Jonath did this job tremendously. And to summarize, how many molecules did we discover over the last, let's say, 80 years? Now it's a little bit surprised. Only 2,500 drugs. I was just mentioning a little while ago that you have millions of purchasable compounds available, and organic chemists are busy making more and more molecules every day. Now they're all coming in millions. Now only 2,500 drugs in the last 80 years. This seems very surprising. It's something doesn't make sense. Does it mean that we don't have any diseases? Well, diseases are definitely much more than these 2,500. And these 2,500 is also a repetition of one disease having multiple drugs and so on. So the summary is that drug discovery pipeline is drying up despite massive increase in genomic proteomic data. Well, what's going wrong? What is it that we can do to rectify this scenario? Now, one issue that you would say, well, most of you have seen this type of flow chart, that it takes about 14 years and 1.4 billion. This has been updated to $2.6 billion. And because it's becoming extremely expensive, so pharmaceutical companies probably are not interested in investing too much money into this because it's a very risky business, and too long a wait and too much investment into a new molecule discovery, right? So this is where I hope that the newer technologies, especially the combination that I was mentioning to you, that chemistry, biology, and IT, can help accelerate the drug discovery process. So in silico interventions are poised to cut down on the cost and time involved in drug discovery. Now, so this is captured by our famous Amitabh Bachchan. If some of you are good singers of this Kishore Kumar song, Aapka Kya Hoga Janabi Ali, well, obviously every problem has a solution. And what is it? Let's rephrase this. Well, we need to find new biomolecular targets. We need to develop new drugs and keep some in store and don't tell microbes you got them, right? That's the general idea. Right? So what's that we are doing at SCL Bio? I will use this word SCL Bio just to show this supercomputing facility for bioinformatics. This is our lab, SCL Bio. So we have been developing methodologies for genome. And this is a novel method based on not sequence level comparisons, but physicochemical properties. So analyzing the genomic data using physicochemical properties of DNA to identify protein coding regions and other functional units on the genome. So that we call as a chem genome. And then once you have identified, let's say, protein is sequence that codes for a protein, writing this mRNA sequence is a trivial matter, and writing using Argobin Khurana's code, genetic code, this polypeptide sequence is also no problem, right? It's very simple. But then comes a big grand challenge, which is how do you go from primary sequence to a tertiary structure of the protein? And this is where we've developed another software called Bhagirath. And this Bhagirath software takes you from sequence to structure. So once you have the structure of the protein, you can also create a structure of the DNA if you want the DNA to be the drug target, or the microRNA if you want, right? Then we are in the business of structure-based drug discovery, creating a small molecule which will mostly inhibit, but sometimes they may activate. We'll talk about it subsequently. So this is a Sanjeevani software that does the job of finding a molecule which will inhibit the function of this protein. We also have the methods for DNA targeted drug discovery. I'll share with you some of the ideas. So the entire pathway now from genome all the way to drug molecule is what we're calling as an Dhanvantari pathway developed at SCL Bio. So from genome you go to the protein, from gene to protein to a drug molecule. I call this as the Dhanvantari pathway. Does this really work? Well, I'll show you an example. So HPV virus is a nuisance. We want to get rid of this HPV virus, right? And what's known about it? Well, there are 4 ORFs, open reading frames, and the function of the proteins expressed by these ORFs is also known. And what is also known is that there are a few FDA approved agents, nucleoside analogs mostly. So due to resistance for these nucleoside analogs, it's expected that the newer drug molecules will be helpful, are essential at this stage, and also other targets are targets other than viral polymerase that is preferred. So what is it that we want? We want a newer target, a different target than polymerase, and some molecule that does not look like a nucleoside. Right? So that's a challenge. In fact, one of my colleagues in the School of Biological Sciences at IIT Delhi, he's a world expert on this HPV, and he said, can you come up with a molecule? I told him very simple, just give me 14 years and $2.6 billion, I'll definitely get you a molecule. Well, more seriously, just give me 3 hours. 3 hours and I'll give you a lead molecule, and I'm sure you'll find that the molecule is working. He says, well, this is a crazy thing. 3 hours for a new molecule, let's see how it works. Now, what do? Luckily, HPV genome sequence is available. So I go to this NCBI website, download the genome onto my laptop, and push this genome onto this ChemGenome. Remember, my ChemGenome software is meant for genome annotation based on physicochemical features of DNA. So what it does is to identify 5 protein coding regions. Now, I could have taken all the 5 protein coding regions, 5 proteins of the virus, because they don't share similarity with the human proteins. But my friend suggested, well, why don't you focus on this pre-core protein, which seems to be very essential for viral replication. So I said, okay, well, let's do this. So we take this pre-core protein sequence. Remember, this came from genome annotation. So once I have the protein, I push this to Bhagirath software. What do? I get a 3D structure out of this software. Now, there are several other softwares, so there is no particular insistence here that we have to use only Bhagirath. Any good protein modeling software does this job, particularly ab initio. Homology may or may not work all the time. So once you have 3D structure, then I'm starting to look for binding pockets. So in an automated mode. So this is where Sanjeevani starts to come in. So this is the drug discovery software. So what it does is to find 10 binding pockets. I could have taken, if I had any biochemical information as to which is the most relevant functional binding pocket on the protein, I could have zoomed in on it. In the absence of that information, I would take all the 10 binding pockets, and I won't miss the true active site, so-called active site. Then I scan a 1,000,000 compound library against each potential binding site using RSPD methodology of Sanjeevani. Remember, this Sanjeevani is a collection of several suites. I will mention a few of them as we go along. Right? Active site finder was one such, and RSPD is another module in Sanjeevani software. So it can scan millions of compounds in minutes, no issue. So after I do this, then I get shortlist about 2,000 molecules. Now these 2,000 molecules are very promising, but I cannot be docking atomic level, all these molecules. So I then put a cutoff and then do a docking. Take all the 2,000 and then do a pardoc. That is again one more module of Sanjeevani with a cutoff. Then I end up with 24 molecules. Beyond this, I go to molecular dynamics simulations. So once I do MD simulations and then I recalculate the binding energies, and I am confident that I have some reasonably good molecules. Well, this is how the story looks like, 24 molecules, and then the job is done. So it's about 3 hours. So what did I do? Well, I did the genome annotation using ChemGenome. I did the 3D structure prediction using Bhagirath. Second step, the third step is to find active site. And after I do that, I do a 1,000,000 compound scan through RSPD of Sanjeevani. And after that, I do a pardoc, that is a docking, atomic level docking. And subsequently, I do on a promising candidates, I do a molecular dynamics with protein and ligand together and individually, and then figure out the binding energies and then shortlist the molecules and then tell the experimenters, can you please test them? So here is one validation. So Vivek was my collaborator. Remember, he's a professor in biology. So Ruchika is my PhD student. So she did this job and we found a couple of molecules. One of them showed a low micromolar, and then surprisingly, it also shows activity against another surface antigen protein. So that's an interesting observation, side observation. But what's guaranteed is a low micromolar compound. Now, this clearly should convince you that with some more medicinal chemistry and with toxicology under control, coming up with a nanomolar molecule is conceivable in an automated mode in the near future. So a couple of things. Medicinal chemistry is required to make it probably nanomolar. Toxicology is required to ensure that it is not harmful to to the organism that's infected, like in this case, humans. So this shouldn't be toxic to humans. So if these two are under control, then you solve this problem of drug discovery, and you can automate the whole process. Now, so this is what I mentioned to summarize to you: genome to some hit molecules using these three software suites and some X teraflops, and of course some smart brain power, that most of you have, right? And now let's proceed to zoom in on Sanjeevani. I will not mention this chem genome, we'll reserve them for some other date. So today we'll talk of Sanjeevani. Now what is this? Well, this is a target directed lead molecule. Earlier I used to call it active site, but now that we are able to find active sites sort of in automated mode, just calling it as target. So the target could be the DNA or a protein, and then we have users sketching the molecule, or a database providing the molecules, and then we have drug-like filters. So Lipinski's filters come in here, then you can do quantum calculations, geometry optimization, some in the force field, then active site identification, target and ligand docking. Then of course there will be a binding and free energy estimate, then followed by MDC. So this is the rough flow chart that we sketched quite some time ago, about 2006 or so. Further on, we moved on, and then we added more utilities to this whole flow chart. So now we have in the Sanjeevani software, we have a non-redundant database of small molecules. We also have a million compound library. We have natural product database. More recently, we have included compounds from Indian medicinal plants. So even they are available. Now, the user, of course, can also do a self-drawing of the ligand molecule. Now, what can we do? Well, if you want, you can check the Lipinski compliance to see whether this is going to be orally bioavailable or not. Alternatively, you can bypass it. So these are all like optional boxes, optional utilities. Now, with millions of compounds, of course, you don't do atomic level docking. So you go through a quick, rapid assay, computational assay screening, and that screening is enabled by this RSPD protocol. And after this, you can go to more rigorous optimization of the geometry, quantum mechanical charge derivation, docking and scoring, and so on. You can also do a DNA sequence to structure A or B DNA form, etc., and then eventually get an MD average structure as well, and then do a docking, scoring, and finally come up with some molecules which are candidates for further experimental testing. Right now, the Lipinski rule. This is one website that's available to you. All you need is to upload the molecule. Now this provides for lots of options. You can provide in different formats, and it'll then give you, well, what's the molecular weight, hyperphilicity, hydrogen bond donors, etc., etc. So the information regarding the compliance of the Lipinski rules are provided by this web server. So what we did was, well, we create a methodology, then we create a web server for free usage, so people can utilize it and then get some meaningful results out of this. The next is the active site identifier, active site prediction methodology, and that's described by this, the URL here, right? It automatically identifies the active site. In fact, this ADS methodology is the next one that I'm going to show you. So but before we release this, what we did was to test this against several popular active site identifiers. When I was mentioning in the example that take 10 binding pockets, the reason was that if you took 10 binding pockets, you don't miss the two active site. But if you took the top one, probably, again in an automated mode. If you had the biochemical knowledge, then of course there is no need. You just focus on only one binding pocket. Now, so the accuracy is a little lower than 100% if it's only one binding pocket that you are focused on a protein with no biochemistry known, and then you're trying to design a molecule. So that's the catch there, right? And then RSPD I was mentioning to you several times already. So this is again a software that screens millions of compounds in minutes. So this is also freely accessible, so you can, some of you can test it, right? BiTalk is another utility where sometimes the function or the bioactivity of a molecule is known, but target is not known. And these are the cases where you say, well, maybe I can do a reverse docking, inverse docking, meaning I have the compound and then I want to scan against several proteins. So we have done against several human proteins, and this website enables you to do that, to find what are the probable targets for a given biomolecule. So this is of interest. So people who know already about some molecules with some bioactivity information. And this is the usual quantum calculation for chart derivation. If you have a new molecule, you have to prepare it for docking, scoring, and eventually doing MD simulations and so on. So this is where these quantum chemistry calculations are essential. Then we started out with docking, rigid body docking. Then we created a parallel version of the docking because this is time taken depends upon the number of configurations that you generate. We have subsequently added flexible docking. We are now trying to web enable the flexible docking version, except the time taken is on the higher side, but the results are very, very accurate. But even with this paradox, the results are reliable as we have seen it through several validations. Now, what does this blind docking do? Well, what blind docking does is to identify binding pockets automatically and then dock the compound and eventually give you the binding energies, free energies. What are the binding free energies? So if it binds it here at any place. So with a threshold, you can say, well, where is the molecule likely to bind? Eventually, you would think that the one with the best free energy should be the one that's most preferred for the molecule. Very simple. And now these are some validations on the docking accuracies and RMSDs versus crystal structures from the top 5 dock structures, some validation studies on the docking paradox, ads programs, and so on. Now, of course, how do you calculate free energy from one structure? Well, obviously, if it's only one structure, and so it's an empirical free energy kind of a function. It's not exactly a free energy because now entropy is missing. So some calibration will be obviously required, right, for this type of thing study. So we created a methodology called BAPL, BAPPL, and that gives you feed the complex and it gives you free energy. And here we are trying to compare with the different methodologies and also with the experimental values, and we find that the correlation is decent, 0.9, etc. And we did something similar for the minor group binders. So you give a sequence of DNA and a drug molecule, then automatically the methodology creates a 3D structure and then scans the molecules against the minor group and eventually finds the site with the minimum most free energy of binding, and then says, well, here is a structure and how does it compare with crystal structure? That's what is being compared here in the second plot. Comparative study of BAPL versus various other popular softwares which were available at that time. Now, of course, I'll mention to you shortly that we have come up with a newer methodology called BAPL Plus, right? And so this is a scenario where metal ions are present in the active site. You have to worry a little bit about such cases. You need to rederive the charges on the zinc or on the metal that's available there because of the charge transfer possibilities. You cannot say, well, zinc is +2 charge and therefore let's just do a calculation using +2. It may not be possible, much less than what you would expect because of the charge transfer possibility. So that's what this BAPL Z software enables you to do. BAPL Z software enables you to do that, right? And now BAPL Plus is published very recently, just a couple of months ago. So what this does is to take any metal ion, not restricted to zinc, right? And this incorporates, not to my taste, but to surprise or to the excitement of my students, they have included AI techniques. I would have been happier using some chemistry force field based methods, but so they have augmented it with AI technologies, particularly random forest. So using these methodologies, they were able to push the accuracies to a much higher level. So BAPL Plus is one of the very accurate methodologies for binding free energy estimates. I will still call them as estimates because the statistical mechanics is still missing. I think that requires, again, a rigorous MD simulations followed by analysis, right? Free energy analysis. And here is another software that does the job of metabolism site prediction, again, developed some time ago. So this is, if one is worried about metabolism related toxicity. We created a molecule, but before it goes to the active site, it just breaks down. Does it really break down into fragments and therefore loses its charm of being the drug molecule? Sometimes it may be useful as a prodrug. So here is the software that tells you that, well, what are the possibilities that this molecule will get metabolized. Now, how about DNA as a target? Now that genomic information is available, so here I am trying to do a comparison of a particular sequence of a Plasmodium, malarial, the genome here, comparing that with humans. How many times does this particular sequence, which is considered to be a reasonably good drug target on the Plasmodium falciparum genome, well, does it occur? Now, if you start looking at this sequence, it occurs millions and millions of times. Remember, human genome is too big, so there is a random chance that it can occur. So when does it not occur? So you're looking for a DNA target that exists in the pathogen, but does not exist in humans, so that there are no side effects. Now you find that if you start scanning, take a sequence and compare that with human genome, you'll find eventually that if you start going beyond 18 base pairs, then the target starts to become unique. Now 18 base pairs is reasonably good drug target for avoiding specificity, but making a molecule that will cover 18 base pairs is a horrendous task. It's just impossible, right? It beats all this Lipinski compliance and several others. Then there are other issues of reaching out to the DNA part. So there are drug delivery issues. What I'm expecting is someday when we get this nanobiotechnology under control and drug delivery becomes a routine matter, probably DNA will be a very attractive drug target. So far, however, proteins are ruling the roost, and especially enzymes in the proteins, because they have a nice pocket, a binding pocket, and a small molecule can do the job of inhibition, and right. But then continuing on the theme of the DNA, this is a comparison of the minor group binders, so it's experiment versus calculated delta Gs and so on. And here is an intercalated methodology for the DNA, because some of the drugs, in fact, are known to intercalate between the base pairs of the DNA, and we now have a methodology which can take care of that possibility as well. And as I was mentioning repeatedly, while BAPL and BAPL plus, those two empirical functions give you a quick estimate of the free energy, you need to at some point, before you tell the experimentalist, synthesize this molecule or test this molecule, you would like to get this delta G naught more accurately using MMPBSA, GBSA, or a free energy perturbation. Now methods are available, several methods are now available to get really reliable estimates, except that these are computationally demanding, but with greater access to supercomputing, this is conceivable and this can be automated, right? Now, what is this here? Well, this is an interesting chart, and that's what I have in mind. Suppose now, someday, all the human protein structures are solved. So I have a data library which has the structure 1, structure 2, structure 3 of all the 20,000 plus proteins, human proteins. And let's say I have created a drug molecule to bind to a protein 1 or a target 1. To ensure that I don't have off-target binding, that is non-targets, what I need to see is that the drug 1 binds to target 1 with good binding energy, but to others with a much lesser binding energy, so that it doesn't bind, or it very loosely binds and it goes away through some equilibrium back to target 1, etc. So to summarize, what I would like to see, this blue color is high binding and orange is a poor affinity. So what I would like to see is blue along the diagonal. So if I'm creating a highly specific drug molecule for a specific protein, and so it's essentially affinity and specificity together, right? So you want high binding, that's the affinity part. You want to be specific too, so that it doesn't go bind to the wrong places, right? Wrong targets. So if you see an off diagonal blue here, like this blue here, drug 13, it is supposed to bind to target 13, but it's binding to target 11, it's binding to target 1. So this is bad news. And now computers can tell you beforehand, before you do the experiment, that there is a possibility that your molecule can go bind to wrong targets. So that's what this story here is. So how do you prevent off-target binding through in silico methodologies before you go to the wet laboratory experiments, right? Now, here are some success stories. Anti-cancer molecules coming out of Sanjeevani. Here, the target was estrogen receptor, and what we predicted using these biphenyl derivatives was that the compound should be micromolar, and it does turn out in experimental assays. When I say success story, I'm suggesting that the computer has predicted, and then they have been validated experimentally, and they're shown to be good binders, at least micromolar compounds are what you have seen so far. So this is again is estrogen receptor. The next story is about anti-Alzheimer acetylcholinesterase was the target, and we had some molecules, and in these molecules, this is pyrimidine, triazole, pyrimidine, etc. based analogs, and they seem to be doing quite well. As at least we are getting micromolar compounds routinely here as well. Now this is an antimalarial and methyltransferase is a peculiar or specific to this falciparum malarial parasite, and therefore that was a good drug target. So we created a few molecules and triazines in particular. So they seem to be doing a good job, and we got some micromolar compounds. In fact, we did end up with some couple of nanomolar compounds, and some further experimental validations are in progress, right? And now, more recently, we addressed this coronaviruses, so 3C proteases, and HIV was a target, and we found that a few molecules seem to be doing a good job meaning micromolar KIs here, 3.3 and etc. We also tested this because of the similarity of the active site of the 3C proteases. We said, can we use this as generic compounds against multiple 3C proteases? Now, I'm sure you can guess easily as to why I'm focusing on this 3C protease, because this nuisance, this coronavirus, does have a 3C-like protease. So this is where this story is going, right? So it does seem to show, these molecules seem to show not only against HIV, against HRV as well, and against a few other computationally, experimentally against these two, computationally against 10 other proteases of this particular coronavirus family, right? So that's a story which just got published last year. Right, and then in collaboration with Harvard Medical School, my friend Arthanari Haribabu, we did have some antifungal, and this is published in the celebrated Nature Journal. And we've been collaborating with the NCI on drug repurposing, and there are several other projects which are ongoing. Now, most of you may like this idea that, well, you can do this drug design, computer-aided drug design, sitting in a metro, or sitting traveling, in a bus, and so on, using this mobile app. So this is downloadable. It's created for Android, and unfortunately not ready for iPhone as yet, but hopefully someday we'll get one for iPhones as well. Right now, where is the drug discovery heading? Well, it seems like we have this affinity specificity under control. What we don't have under control is the T part. The ADME part seems to be under control, the T part. So if you can give me 5 rules of toxicity, well, imagine the drug discovery is automated. Now, most of our expenses, when we need to worry about these various phases of trials, etc., because of this failure rate is too high. And if somehow you can prevent this failure rate, focusing on this toxicity, just like the Lipinski rule of 5, you can come up with a toxicity rule of 5. Well, that'll be wonderful. It's a new dawn of drug discovery. So hopefully someday this will happen, right? So I mentioned to you in silico drug discovery pathway, and we have tightened this whole pathway, and then just pushed it into this chemical engineering science more very recently, just a couple of months ago. So from chem genome to Bagheera to active site finder, RSPD. So these 3 are parts of Sanjeevani, RDOG, and eventually some MD simulations and small molecules, and hopefully ADMET filters. Now, most of you are interested. Well, if you have a drug design software like Sanjeevani, why not do something about it against COVID-19, right? It's a pressing need, right? And here for some information, and this is available all over the place, so I'll skip this part just to tell you that there are several proteins here. Some of them are structural proteins and some of them are non-structural proteins. NSP's non-structural proteins are good drug targets, and particularly enzymes. So we found that these three are good drug targets: NSP5, 12, and 16. Now, what do these do? This is a 3C-like protease I was mentioning to you a little while ago, and this RNA-dependent RNA polymerase, and then there is a methyltransferase as well. So they're luckily, structures are available for this, so you don't need to model them. So PDB IDs are provided here. So how do you then do? Well, we did 4 different things. So we took FDA-approved drugs. Remember, there are 2,500 molecules which are already reported, and new molecular entities. And then there is a drug bank which can give you more details about it. So we can take all of them and scan against each of these proteins, the binding pockets of these proteins. And then what do we end up with? Well, so what are the steps involved? The steps involved are, of course, the scanning of the molecules and then docking of each molecule. Because this number is small, you can go for docking each molecule. You don't need this RSPD type quick accelerated method of screening. So then shortlist the molecule, then perform MD simulation, then use a more rigorous free energy calculations and evaluate the molecules and say, well, here are the proposed compounds. So that's one set of input molecules coming from FDA approved drugs. What about another possibility? I was mentioning to you a little while ago that against HIV, we found some molecules which are generic 3C protease inhibitors. And these are isoteins. One of my colleagues, Professor Ravi Singh in the chemistry department, he is an expert on the isoteins. He routinely creates them. So we are testing here molecules which have proven to be good macromolar binders to this HIV. So again, we tested this against the 3 targets that I mentioned to you for this SARS-CoV-2 proteins. And we have some potential isotein molecules as well. Now, Indian medicinal plants. In fact, we have focused on tulsi, neem, and turmeric, and we identified the compounds that are available there. And then we created a website called Bioactivity Informatics of Indian Medicinal Plants. So this website is freely accessible, and you can download the structure. You have lots of links there telling you what these molecules can do, etc. And what is their bioactivity. So you can take these molecules and then process them against the 3 proteins that I was mentioning to you of SARS-CoV-2, right? And then we have some potential candidate molecules again binding to this. So that's the 3rd stream of molecules. The 4th stream, of course, is this so-called Zinc database, which has millions of compounds. So this is where you will use this rapid screening methodology followed by a detailed atomic level docking scoring and MD simulations and so on. So we have some hit molecules. So where are all these molecules going? Well, these molecules are now waiting for further testing. Initially, in collaboration with IICT Hyderabad, we have done protein binding assays, and they seem to be showing good promise. And now we moved on to NIV Pune, National Institute of Virology Pune, for BSL-4 level testing for viral clearance. And I hope that the results will be encouraging so that we can proceed further, right? So that's where the application is of these molecules. And again, thank you very much for your attention, and we'll be happy to take any questions. And if you have some questions, you can always email them to me, and I will be happy to answer. Thank you.
Sir, thank you, sir.
Thank you, sir.
Yes.
Sir, we have received some. Just 1 minute, sir. While your talk is going,
yeah,
we be able to receive some questions from YouTube channel. So I just will pick up some 3. I picked up some 3-4 questions.
Yeah.
So, sir, actually there are some general questions like this Sanjeevani software, when we are submitting this calculation, whether it will run in your server or it will consume our system power or consume all this information.
Yeah, one quick answer to that. So these are all web server mode, and they work on the dedicated SEO biomachines. So some processors are dedicated. For example, Pardoc, it consumes 80 processors. So 80 processors of this SEO bio are dedicated for taking on the various jobs in the queue. So user can use a mobile phone or a laptop, and it doesn't consume power from there. The computing power is coming from SEO bio. So that answers that question.
Yeah, thank you, sir. And one more student, he is asking, I mean, what is the algorithm behind this Bhagrit as well as that Sanjeevani works?
Yeah, so yeah, let's go. I think Bhagirath we'll save for some other day because that's a protein folding, protein structure prediction algorithm. So the Sanjeevani part, there are several steps. I showed you a slide where several steps are involved, right? So if I can go back to my slide. No. So overall, oops, I have to skip this slide. I'm sorry for this.
Yeah, no problems.
So this is the algorithm.
Sir, can you please share your share? Yes.
It's not shared?
No, sir, not yet, sir.
Where do I do this? I think I missed that. Where is your, okay. No, is this visible?
Yes, sir. Yes, sir. Now we are able to see.
Okay, I'll go to that flowchart side. So this is basically the flowchart. So we have a little software for each of the steps involved, including this retrieving the molecules from this or converting the molecule to a particular format, pushing them to drug-like filter, and for the optimization, and then QM derivation active. So each of them has a little software which is published separately. So if one visits these papers, and so one gets the detailed knowledge. The overall flowchart is what I'm showing, and a more detailed flowchart is there in the next slide, which has several things like this databases with the molecules that are getting input here. There's a protein or the DNA sequence, and then force field parameters, docking, scoring, and so on. Right. So those are. So it's not just one algorithm. It's a collection of several software tools, which is collectively called Sanjeevani. All right. Any other question?
Yes, sir. Sir, in your case study for against the HBV, there you have mentioned like finally you have screened 24 compounds from the million compounds.
Oh no, the 24 compounds is what came out, and so coming from million to 24 is a big thing, right? So I'm sure all of you will appreciate. So and then finding that the molecule is micromolar binder is an exciting thing, definitely, right?
Yes.
And that's to my assessment. It's not just Sanjeevani, that is where the CATD methods are. I believe that the computer-aided drug discovery has come of age. So today, computational chemists, in collaboration with, of course, biologists, etc., can easily come up to a micromolar compound from whatever the input library, million compound library, or medicinal compound library, or natural compounds, etc. Where are we lagging behind in this CATD? Well, we are still unclear on the areas that contribute to toxicity. Therefore, that's a severe bottleneck. The second is automating this micromolar to a nanomolar. So that automation requires a molecular level intervention. I probably have to put a fluoro group or a methyl group or etc. So that is where intuition of the organic chemist has to be captured into an algorithm, right? So those are the areas where one can invest time in for ensuring that CADD never fails. Right. So today we are sure, whether it's a Sanjeevani or other softwares, and there are some commercial softwares which are available. Utilizing any one of the state-of-the-art softwares, and one can easily go from the target to biomolecular target to a micromolar compound, making them nanomolar, ensuring them that they are non-toxic are the next steps in this area of research. So that's really my observation.
Thank you, sir.
Yeah.
Sir, one last question. So one student, she just asked, like, what is the difference between reverse docking and inverse docking?
I used it here colloquially to mean the same thing. Essentially, there is a software called inverse dock, which does the job of finding the target given a molecule. In the regular docking, you'll find a molecule given the target, right? So I'm using it in a loose sense.
Yeah. Oh, thank you, sir.
Colloquially, right?
Thank you. So with now we are coming to the end. So with this, on behalf of Drug Discovery Hackathon 2020 Training Committee Chairman, Dr. G.N. Shastri, I would like to thank you for your presentation as well as interaction with, for the question and answer session. So now we are going to end in this particular session now. So, and tomorrow we will have the question and answer session, and we will announce in our website by tomorrow morning. Thank you.
Thank you very much. Thank you very much,
Professor
Jayaram.
Yeah, thank you, Professor Shastri. Thanks a lot.